think, well, we've got quite a lot of people online already, so do you want to start? And everybody put your questions into the chat and we'll go to the to the questions at the end. So Malcolm, do you want to start when you're ready?
Okay, thanks Rachel. As people may have heard, as we're setting up, the internet connection isn't terribly good, so I hope that you can hear me throughout. I'm going to talk about our work on redox, which is redox, free radicals, reactive oxygen species. These are things that we've been interested in for a long period of time. As you've seen from the title, skeletal muscle responses to exercise and aging. I'm going to talk about the work of the group rather than my own, just my own work. And there's a lot of people that have been involved in this. Just a few weeks ago, Anne McArdle presented the lunchtime seminar, and she gave quite an overview of what the muscle group were doing. And I thought I'd do something a bit different and be much more related to a very specific area, and that's the redox regulation of responses to exercise and how they're modified by aging. When I talk about redox, I'm predominantly talking about hydrogen peroxide, so I'm afraid you're going to hear a great deal about hydrogen peroxide today. So, okay, so one of the things we've been interested in for a great deal of time is how muscle adapts to contractile activity. We've spent a lot of time looking at the very rapid adaptations to contractile activity, which we demonstrated were redox regulated a number of years ago. And the slide that you've got on the left-hand side shows some studies from mice, which were originally done by Aphrodite when she was a postdoc in the lab. And then on the right-hand side, some studies from humans. They're looking at a series of rapid response proteins, heat shock proteins, some of the antioxidant enzymes and so on. The message here is that if you look at young animals or young people, then you get these very rapid responses to contractile activity. But if you look at older animals, those responses are attenuated. We were the first to describe responses such as this, which are redox regulated. It's now widely accepted that a whole series of other things like microbiogenesis, substantial parts regulated, and you get things as well, and a number of other areas. And so we've become interested in quite a wide-ranging group of adaptations, which are in part stimulated by generation of free radicals, reactive oxygen species, during contractile activity. So this is my plan. I'll talk about hydrogen peroxide and then very much about peroxyredoxin signaling in response to contractile activity. I'll then move on to talk about what happens with aging and the role of motor neurons and the innovation in muscle mitochondrial redox homeostasis. Then we'll come on to a new concept that we've been talking about, which is compartmentalization of redox, particularly hydrogen peroxide signaling in skeletal muscle, and then try and summarize that at the end with a hypothesis. So this is a slide which we've been working on for a number of years, which shows the potential sources of reactive oxygen species generation in skeletal muscle. I just use this as to get us started, really. There are only, in fact, two of these species which are generated by skeletal muscle. One's superoxide, which can be generated both in mitochondria and in the cytosol, and the other one's nitric oxide. And there are a number of compounds which are substances which are derived from that. The most important, as far as we're concerned, being hydrogen peroxide in the mitochondria, or within the cytosol, and actually outside the cell as well, as you can see here. So hydrogen peroxide seems to be the key signaling molecule which is involved in this redox regulation. And this is what we think goes on. The things I'm going to talk about, initially at least very much, are around the concentrations of hydrogen peroxide that you expect to find in a muscle fiber. In the resting muscle fiber, the predominant sources of the hydrogen peroxide that you find is actually coming from outside, because extracellularly, hydrogen peroxides are around 10 to micromolar, whereas intracellularly, and this is the big surprise, the concentrations are in the nanomolar level. Okay, so what happens with contractions? Basically, you activate an NADPH oxidase. The subunits assemble on the either the plasma membrane or the T-tubule membrane and generate superoxide. There's a local area of high hydrogen peroxide concentration generated. Some of that comes back into the cell where it slowly diffuses and decreases in concentration. That elevates the intracellular hydrogen peroxide by about 100 nanomolar as far as we can see. These are incredibly low levels of hydrogen peroxide. And that's really what one of the puzzles that there's been over a number of years. Because actually what people were assuming was happening was that these peroxides which were generated intracellularly were directly oxidizing key components of some cell signaling pathways. And the ones that we've been interested in which people have reported there's activation by hydrogen peroxides are things like the MAP kinases, the protein tyrosine phosphatases, the mitochondrial transcription factors such as PPAR gamma, and of course NF-kappa B. When you look at the literature, actually for MAP kinases, for instance, and for PTP1B, then you require something like 200 micromolar hydrogen peroxide to activate those by oxidation. And that's similar for IKK in the NF-kappa B pathway. So there's a big mismatch between the concentrations of peroxide, which are really being generated within the muscle pathways, which are said to be important. And it's quite interesting because you can use antioxidants to demonstrate that oxidation is playing a role in the activation of these pathways. So there has to be some link between the generation of the peroxides and the activation of the transcriptional pathways. So we've hypothesized for a number of years that there needs to be something sitting in between this peroxide production and the activation of the pathways. So what can react with hydrogen peroxide at these very low concentrations found in muscle fibers? And the answer is peroxidases. So this slide here shows a number of the compounds on the left-hand side which can actually react with hydrogen peroxide, things like the glutathione peroxidases, the peroxiredoxins, and the thioredoxins. And this is a paper from Cell Reports in 2017, which is quite interesting. It was pointed out to us by a journal. Usually we hate reviews of journals, but actually this reviewer gave us some quite interesting information and suggested we look at the relative concentrations of these different compounds that could react with the hydrogen peroxide. And you can see that the peroxiredoxins really predominate. This is mass spec data, so these are relative absolute concentrations, relative concentrations of these proteins as they're found in the muscle fiber. You don't really get this information from Western blots, of course, because you can't really compare between different antibodies. So peroxiredoxins and these other peroxidases are really interesting to us. And Christine Winterbourne in New Zealand has done some really interesting work where she's tried to compare the reactivity of these different proteins. So for instance, this tyrosine phosphatase 1B, which reacts with hydrogen peroxide, but it has a relatively low capacity for reaction, and it isn't very reactive. Glutathione, which is present in cells at up to millimolar concentrations, but doesn't really react terribly well with hydrogen peroxide. And these peroxiredoxins, which have got this tremendous ability to react with hydrogen peroxide. And she's calculated the percentage of the hydrogen peroxide that you anticipate within the cell, which was actually reacting with each of these components. And you can see that she says effectively, she finds effectively that peroxiredoxins are reacting with almost all of the 100% of the hydrogen peroxide you'd expect to find in the cell. Whereas these signaling pathways actually don't, these signaling molecules actually don't really react at all. So there has to be something which is, come back to what I was saying a moment ago, which is sitting between the hydrogen peroxide generated and the signaling proteins. And the answer to that seems to be peroxiredoxins. So this is a figure from a slide, which is it's actually a figure from a paper which has just been accepted for free radical biology and medicine. Claire Stretton is the first author. And this is actually, it's quite complicated, but I'll run through it. It shows the reactions of hydrogen peroxide with peroxyredoxins here. So peroxyredoxins contain these two cysteines, and these two cysteine peroxyredoxins highly reactive, as we've just said. What can happen is they can be, at the concentrations and low nanomolar, they can actually form sulfonic acids. These sulfonic acids then can further condense to form peroxide disulfides, dimers, and then you do these disulfide bridges, as you see here. So this is the way that actually peroxyredoxins act as peroxidase. And the most likely thing that will happen to them is they'll very rapidly be reduced back to the monomers by interaction with thioredoxins. And eventually that utilizes NADPH. That's the peroxidase activity of peroxyredoxins. But of course, once you've got this dimer formed, then that is itself an oxidizing molecule. And it can potentially, by disulfide exchange, pass on that oxidizing potential to other target proteins, other proteins that contain these thiol groups. And that's been shown to happen now for 3 different proteins. And this is it's been called a redox relay, because you're actually passing this, this redox potential onto another protein in the formation. And these can then exchange with key signaling molecules that are involved in this. And that's one of the things that we're trying to identify as things slightly more. The other thing that happens is that if you've got too much hydrogen peroxide there, the peroxyredoxins become hyperoxidized, so they've got the sulfonic and sulfinic acids, and they are then non-reactive. So they are actually steers the dimer, they can't go through this pathway at all. And if you look back at the older literature, certainly prior to the 1990s, there's no mention of peroxyredoxins. They were thought to be completely unimportant. And the reason is that most people at that stage, and I suspect most of us, are using hydrogen peroxide in our experiments at micromolar level. And in that situation, you completely remove this pathway. This is the only pathway that occurs to become functional. So it used to appear that peroxyredoxins play no role in hydrogen peroxide metabolism at all, but it's only that they don't play no role because they're oxidized once. So the key thing then for going forward is that you can demonstrate this dimerized form and the monomers using a non-reducing gel, and it's a straightforward approach. And that's what we've been trying to do, looking at muscle contractions. So this is using FDB fibers, isolated FDB fibers, and in culture we can stimulate these to contract. And if the reason we use these is because it provides very ready access for allow us to get alkylating agents in to prevent the spurious oxidation of peroxyredoxins. And what you see as you move from the monomer to the dimer, this is actually the oxidation of peroxyredoxin occurring at lower hydrogen peroxide concentrations. And so what we've got here is a stimulation protocol of half a second every 5 seconds. We stimulate these FDB fibers for a minute, 2 minutes, or 15 minutes, as you can see. And actually what happens, we've demonstrated for the first time this oxidation of peroxyredoxins. Now, the peroxyredoxin 1 and 2 are both found in the cytosol, and peroxyredoxin 3 is found in the mitochondria. So in the cytosol, we get an oxidation occurring in peroxyredoxin 2 within 2 minutes, peroxyredoxin 2 within 1 minute, peroxyredoxin 1 within 2 minutes. And actually, we're also seeing mitochondrial oxidation of peroxyredoxin 3 occurring within 2 minutes. I'll bring you back to the contractions. There's actually 12 contractions. So within a very low number of contractions indeed, we're actually seeing this oxidation occurring. So if it's a real system that's playing some role in situ, it would also be reversible. So that's shown here. So again, we've got this movement from the monomer to dimer. The data you've already seen from peroxyredoxin 1 is oxidation this time to 15 minutes. But then what Claire's done here is left the muscle fibers to relax for 15 minutes, 30 minutes, 120, and you can see that the process reverses. And it reverses for all of the 3 peroxyredoxins that we've actually looked at. So what do we think is going on? Well, now think that we're seeing this link between the formation of hydrogen peroxide and the activation of the transcriptional pathways. And that's this so-called redox relay, this oxidation of peroxide. The key next thing to sort out is what are these target proteins. And so what we've been doing here is Claire's using a trapping mutant system to allow us to try and look at that with mass spectrometry. We haven't got any of those yet. That's a complicated part of it. But that's really where we looked at. And I'll come back later on in the talk to what happens to peroxide oxidation during aging. So that's part one of the talk. I want to now move on to talk about aging and the role of motor neurons and denervation, and particularly their effect on mitochondrial redox. I remind you that aging attenuates these responses that we see both in mice and humans. So there's lots of evidence of the aging. These are some various different bits of evidence from our work. The most obvious one is increase in oxidative damage. It shows some carbonyls increasing in old animals, and also things like malonaldehyde. That's thought to be due to an increase in the mitochondrial generation of hydrogen peroxide, and this is data that Aphrodite Vasilaki did with Holly van Remmen back in 2006. And this is looking at peroxide production from isolated mitochondria, and you can see that it's elevated in muscle from old animals. We've done some work with non-specific probes, such as DCFH, and you can see that DCFH oxidation increases with contractions, but in the old animals, it's actually already elevated before we do any work with contractions. And then indirectly, there's lots of evidence that things which adapt to reactive oxygen species, regulatory proteins, things like catalase and GPX that we've talked about before, and particularly thyrodoxins, are elevated in the muscle of all animals as well. So there's lots of evidence for previously published in this area. So I just want to tell you a little bit about some work we've done now over nearly 12 years with this group in the US under a National Institute of Health, National Institute of Aging program project. Several people have heard me talk about this in the past. I just want to bring you up to speed and tell you about the latest work in this area, because it helps in terms of our understanding of what's going on. This is the slide you've already seen of the potential sources of reactive oxygen species in muscle. And what we've done with this US group, or what they've done, is actually knocked out various different key proteins to see whether we could manipulate the rate of aging by modifying the generation or the activity of these species. And the things that they did, we started off looking at manganese SOD. It's very damaging to the cell, but doesn't seem to be associated with aging. We looked at catalase and glutathione peroxidase. There was no real interest in phenotyping those animals at all. We looked at extracellular SOD. There was no interest in phenotyping those animals. We looked at various different aspects of the neuronal nitric oxide synthases, and again, not really an interesting aging phenotype. But when we looked at copper zinc SOD, or SOD1, what we found was an interesting muscle aging phenotype. So basically, SOD1 null animals have a premature muscle aging type phenotype. And this is the original article from Florian Muller at that stage in University of Texas, showing this accelerated loss of muscle mass with aging. We have studied these animals extensively over a long period of time. The most obvious thing that I want to lead you into is a demonstration that this loss of muscle mass is associated with changes in the nervous supply, the neuronal supply to the muscles. That maybe isn't a surprise, because the SOD null mice, of course, or SOD1 mutant mice are associated with motor neuron disease. But when we took these mice and looked at their muscles and then their nerves, we found some quite interesting changes. So we're looking here at neuromuscular junctions. For those who haven't seen these before, they're normally looked at using microscopy. You can demonstrate the acetylcholine receptor on the muscle using blood growth toxin staining. You can stain up the motor neurons coming in various different ways. We tend to use a transgenic mouse to allow us to do this. And then this is the oval layer showing the neuronal input to the acetylcholine receptors, the the synapses as such. And when we looked in the SOD null mice, you could already see there's disruption of the acetylcholine receptors, that the axonal input is disrupted. And then when you see the merge down here, there's quite significant changes going on. You should say this is actually very patchy across the muscles of these animals. How that relates to what happens in old animals as well. And so this is an old wild type mouse with a similar situation. And again, patchily across the muscles, you can actually see disruption of the acetylcholine receptors. This one's got huge disruption of the axonal input, and the merge looks very disturbed. We want to know if that actually happened in humans as well. It's really quite difficult to find, but actually if you look in the literature, there are 1 or 2 post-mortem studies where they've managed to look at the neuromuscular junctions in older people. When you think about it, that's a horrendous thing to try and do. Trying to find in a human muscle fiber where the neuromuscular junction sits is really very difficult indeed. But actually, so these are the techniques that they were able to use back in the 1980s. So this is acetylcholine esterase staining. It's this nice compact acetylcholine receptor. This is actually bungarotoxin staining using iodine 125, and this is silver staining to look at the actual nerves. And you can see that this is spreading very much like you see here in the animals. So the acetylcholine receptor is spreading, and there's branching of the nerves coming into the neuromuscular junction. So the same thing happens in humans as well as we get older. So this is just a summary of a lot of the work we've done on these animals. As we said, the whole body mouse reiterates the effects of the aging on motor neurons and on muscle. The first thing we thought we'd try and do was look at a muscle-specific knockout. That was the obvious thing. To our surprise, it had virtually no phenotype at all. So the SOD1 null knockout really isn't doing very much in the muscle. So we thought, okay, so it must be the nerves that's doing everything. So we did a couple of things. We actually looked at a neuron-specific knockout, and that was also found to a very minor phenotype. But then to our surprise, what the San Antonio group did was actually took the whole body mouse and they rescued the nerve by putting a human SOD1 back into the nerve, and that actually prevents the whole of the phenotype. So it prevents the muscle loss and atrophy and prevents the neuronal changes as well. That's pointing us towards the nerve. But we couldn't really understand why we couldn't demonstrate an effect of a neuron knockout. And when I spoke to the Transgenic Club last year, that work was still ongoing, and there was a lot of debate about exactly what promoters we were using and so on. And I just wanted to update you because we've now completed that work, and we've got an inducible motor neuron specific knockout, and we've been studying that over the last year or so. This paper's in Aging Cell in Revision, and this is actually the key data. So this is the isometric force from the gastroc muscle of both these wild type mice and these inducible, this is a tamoxifen inducible motor neuron specific SOD null. And you can see by 16 months of age, they're induced from about 4 months of age. We're getting a suggestion of a fall in isometric force. But out to 24 or 26 months, when the wild type are just starting to show changes in isometric force, we've got a massive decline. If you look at specific force, you can see that you're seeing exactly the same pattern. So there is an effect, there is a phenotype, there is an effect which is really an accelerated aging phenotype. It takes quite a long time to develop. The reason for that seems to be, one of the things leading to this is shown here. So what the now Oklahoma group have been trying to do is to count motor neurons in the ventral horn. And what they've shown is that by 10 months of age in the inducible mice, we've got about a 25 to 30% loss of motor neurons. Remember, this is associated with very little effect on the muscle force generation. That's about the same loss that you get in wild type control mice by about 18 months. But by that stage, the inducible mice have already lost a lot more motor neurons. And there's changes in the motor neuron area as well. So what it's looking like is that there's a very early change in in motor neurons, a loss of motor neurons in these animals because of the lack of SOD. But the animal is able to compensate by that at the muscle level, probably by expansion of motor units. This is some work that Natalie has been doing with these animals as well. Again, we've got these acetylcholine receptors, the axonal input, and then the merge here. And what we've actually got, adult wild type, we've got old wild type, and we've got the inducible motor neuron knockout here. And you can see that actually at this stage, we can see lots of this magenta staining, which is the acetylcholine receptor coming through. And these are denervated and non-innervated acetylcholine receptors. And Natalie's preparing a paper in this area at the moment. It's quite a complicated graph down in the right-hand corner. But what she's looking at is that she's counting innervated neuromuscular junctions, partially innervated neuromuscular junctions, and denervated neuromuscular junctions. So in the adult wild type, almost everything's innervated. In the wild type, we've got some partially denervated, and but very few which are fully denervated. There's the whole body SOD1 knockout, as you can see. There's actually about a third of the motor neurons, the neuromuscular junctions that we look at are innervated partially and denervated. But the inducible, we don't get this big increase in the partial and denervated until late on in life. So it's this, the inducibles are showing the phenotype, but they're only showing it in accelerated form later on in life. So there's something else going on, which is preserving the muscle structure and the muscle function until that late stage. Okay, so this is really summarizing now the whole of the story. The inducible neuron is an early onset of sarcopenia, about 22 months. But the implication of all of this is that the redox status, the neurons, is particularly important in regulating muscle phenotype. So the paper that we've become more and more interested in over the years again comes from this guy, Florian Muller, who produced the SOD null mouse paper a while ago. And at the same time, he produced a really interesting and very simple paper where all he did was actually cut the motor neuron. Well, he cut the nerve, he cut the nerve innervating a muscle. And what he used was the peroneal nerve, cut the peroneal nerve, looked at the tibialis anterior muscle. He then isolated mitochondria from the muscle, and he looked at mitochondrial peroxide production, which is in this top right-hand graph here. And what he saw was this enormous rise in state 1 hydrogen peroxide production. This is the biggest rise anyone's ever described in mitochondrial peroxide production, all due to the denervation of the muscle. And we thought this was really interesting and was bound to link back to what we were seeing in the SOD1 null mice. And so we've done quite a lot of work using this sort of system to try and understand how the nerve is actually regulating muscle mitochondrial peroxide production. So this was our hypothesis. Actually, the age-related changes in neuromuscular junctions, I said before, it's patchy. And this is what you see if you look at the mouse tibialis anterior. You can see the neuromuscular junctions in a Thigh1 YFP mouse. And there is only some of them which are showing denervation, as you can see here, and some which are partially denervated, as you can see here. So it's in a minority of the fibers with showing full denervation. And so our hypothesis that we're working on was actually that if an occasional fiber was showing denervation schematically here, then the mitochondria in that fiber would start generating hydrogen peroxide, and that would potentially diffuse into neighboring fibers and affect their redox status. So what Natalie Pollock did was some really elegant work where she actually started to transection of the peroneal nerve and actually then looking at what happened to the muscles. And so this is her nice 3D image showing the nerves coming in and the intact neuromuscular junctions on this muscle fiber here. She cut the nerve, an innervated nerve, and this is what you see after 7 days. The innervated nerve by Müllerian degeneration has actually disappeared, and all we've got left is the acetylcholine receptors. This is what happens to what fiber atrophy over that time. And this is what happens if we then take bundles of fibers from the denervated muscle, we permeabilize them, and we look at mitochondrial peroxide production. And you can see we get this huge rise, same as Florian Muller did, in mitochondrial peroxide production. So that's an interesting model, but what would be a much more interesting model would be able to, if we could have a muscle where we got some of the muscle fibers which are denervated and some of the muscle fibers which would retain innervation. And what Natalie was able to do was actually to produce that model by transecting one branch of the peroneal nerve. And so this is actually her peroneal nerve coming into the tibialis anterior muscle, which is shown here. And there are three branches, in three, just before it enters the muscle. And you can see that here, Natalie has actually transected this one, and it's starting to degenerate. What that produces is a patchy pattern of denervation across the muscle. And so what we got here, Natalie was able to separate out the muscle into these regions. And in region 1, we've got full denervation. Regions 2 and region 4, we've actually got partial denervation. And in region 3, very substantial region 3, which you can see here on this schematic, we've got fully innervated muscle fibers. So she took bundles of fibers from each one of these different regions, and we assessed the mitochondrial peroxide production. And what you can see here is that, to our big surprise, all of these bundles, all of these regions have got an elevated peroxide production. So this denervation is not only affecting the fibers which are immediately denervated, but it's also causing responses in the neighboring innervated fibers as well. And so our hypothesis, whereby we were thinking we actually see this diffusion across, actually what we're seeing is that then oxides as well. This was a big surprise to us, as I said. Part of our trying to understand what's going on. So we've talked about what happens with denervation and mitochondrial redox homeostasis. And I want to introduce the idea of compartmentalization of the redox signaling and the redox peroxide sources within muscle. And this brings us on to some work that Caroline did when she was working with us. And what Caroline was doing was actually it's been a bit of a great to try and get a probe which would signal hydrogen peroxide levels, and we could actually express that in vivo. And the probe that we used at this stage is a probe called HYPER. So HYPER is the bacterial transcription factor OxyR, which is responsive to hydrogen peroxide. But what the group that developed this probe did was they inserted this YFP, circularly mutated YFP, into the OxyR, so that once it reacted with hydrogen peroxide, you have this disulfide bond being formed here, but you would also activate the fluorescence from the YFP. Luckily, it turns out to be a ratiometric probe, and you see the changes in fluorescence that we see here. We expressed it in mouse muscle using an AAV, so adeno-associated viral vector, and you can see the arrangement that Caroline was using here, so that she was able to actually put the muscle underneath the microscope of our confocal microscope. She did some experiments just initially to see that we were responding to hydrogen peroxide. That's shown here. This is a huge response that we're seeing to, I think, quite a substantial amount of hydrogen peroxide, 250 micromolar. And then we set about trying to understand what was happening to hydrogen peroxide in muscle fibers. I should say this is a cytosolically localized hyper, because being genetically encoded, you can actually encode it in you can actually locate it in different parts of the cell. And what we found was that looking across these 50 odd fibers in one muscle, that we actually got really quite tight 488, 405 excitation ratio. I mean, the hydrogen peroxide was quite tight. And when we looked across 5 TA muscles, there really wasn't that much difference going on. So what Caroline did was a slightly different experiment, where rather than cutting the nerve, she actually crushed the peroneal nerve. And when you crush the peroneal nerve, what you see here is after about effectively very little of the nerve left. And by 7 days, all you can see is the acetylcholine receptors. I should say that there's a mice, the thyroid CFP, so cyan fluorescent protein. That's why you get this different color here. And that's so that we can also express in these animals the YFP of the hyper. But by 21 days, we've seen substantial recovery. So again, she took bundles of muscle fibers and looked at mitochondrial peroxide production. And at this point here, 7 days, just like Natalie found with the full denervation, she's getting very significant amounts of peroxide being generated from mitochondria from the fibers in the muscle where we've got nerve crush. And that's reversed by about 21 days, as you can see here. So what was happening to our hyper? So we know what's happening in the mitochondria, which is shown what's happening in the mitochondria. It's actually, we've got big increases in mitochondrial hydrogen peroxide production. And when we looked at the mean hyperfluorescence across the fibers, the 50 fibers that you could see in each of the muscles, then what it looked like was very similar things were happening. But actually, in reality, it wasn't what we were finding, because now we can look at individual fibers. We can decide what's the hydrogen peroxide concentration of each one of these fibers. Only a very small proportion of the fibers were having very large rises in cytohyperfluorescence. So in other words, only in very few of the fibers was there a big increase in cytosolic hydrogen peroxide concentration. So Caroline went on then to look at what was happening in old animals, because you know in old animals we've got a big increase in mitochondrial, muscle mitochondrial peroxide production, as you can see here. But when we looked across the board at the cytosolic, these are individual fiber hydrogen peroxide concentrations. You can see that there was actually no significant difference. This suggests that maybe 1 or 2 fibers might be slightly elevated. We'd assumed that what we were going to see here was that each denervated fiber would actually have a big increase in cytosolic peroxide concentration, but that doesn't occur at all. And so what Natalie's now done is done the same thing, looking back to our SOD1 mice, our knockout mice, our model mouse. And you can see, again, this is the cytohyperfluorescence again in vivo. And you can see what we're seeing here is exactly the same thing again. Maybe 1 or 2 fibers showing an increase, but most of the fibers don't show an increase in cytosolic hydrogen peroxide concentration. So what's going on? So what we originally were hypothesizing was this acute loss of structures, loss of energy, would lead to this large increase in muscle mitochondrial peroxide production. We'd already shown from Natalie's work that this propagates to neighboring fibers, and we'd hypothesized that actually, rather than being damaging, this was potentially a process that was meant to stimulate repair pathways. And there's really quite substantial evidence that peroxides, for instance, or hydrogen peroxide, can actually stimulate axonal budding and axonal growth. And so that made some sense to us. I'll come on a moment to talk about stimulation of redox adaptations. What we had assumed would happen was that it would also lead to a big increase in cytosolic peroxides, and that's what would be damaging to the muscle fiber with the disruption of muscle proteostasis and activation of atrophic pathways with loss of muscle mass. But actually that doesn't happen. And why doesn't it happen? Well, it does seem to be due to compartmentalization of the hydrogen peroxide. And it's almost certainly just due to the activity of cytosolic regulatory enzymes, things like thioredoxins, perhaps peroxiredoxins, and so on. We started working on this in which we were aware, and actually as we've been doing this, other people have also suggested that there's some compartmentalization going on. And fortuitously, Richard and Caroline and myself have actually got a grant from BBSRC to allow us to look at the transfer of hydrogen peroxide across membranes. When we got that grant, I hadn't realized it was potentially going to be quite as important as it appears to be from the data that we're getting at the moment. So these adaptations that occur, of course, I showed you this graph before, lots of them occur in particularly these changes in redoxins, but also peroxidase and catalase and so on. And we suspect that's why you don't see this increase in hydrogen peroxide in the cytosol of any of the fibers in aging mice. The denervation itself induces the ones we're looking at here due to the peroxide which is being generated. And so this is some work that Matthias Scalabrin, who's a PhD student who just finished with us last year, Aphrodite was the senior author on this paper. And this shows that peroxidases are going up very dramatically within this 7-day period, as do thyrodoxine reductases, no effect on superoxide regulations from MNSOD, but we weren't expected. And also some of the heat shock proteins, these stress response proteins, are showing very dramatic and very rapid and very dramatic changes in content over a period of time following the denervation as well. We begin to think this is actually key to what might be going on. So we've talked about these first 3 aspects. We've been talking about compartmentalization of redox signaling. And I said I'd come back to talking about what the effects of aging on redox signaling. It won't surprise you that the work that Claire's been doing, we also looked at the effect of age on the peroxidoxine oxidation. And this is the particular graph from the paper which has just been accepted in FRBN. So we look at the mitochondrial peroxidoxine, mitochondrial peroxidoxine 3, you can see that there's very little difference between fibers from old animals and fibers from young animals. They behave very similarly. They both show an increase in oxidation with time, and there's no difference at rest. If we look at the cytosolic peroxidoxins, you're seeing something very different indeed. So first of all, there seems to be an attenuation of responses, which fits with this attenuation of responses of redox-regulated adaptations that we've seen. But also, if we look at rest, you can see that actually there seems to be something different going on. There's a reduction in the amount of peroxidoxin 2, and there's peroxidine 1. And for looking at peroxidoxin 2, there's already a significant oxidation, as we said, by 1 minute and 2 minutes in peroxidoxin 2 in the adult animals, which you don't see in the older 2. But coming back to this change at rest, so I've just brought those out here. Peroxidoxin 2 is showing a reduced oxidation at rest. So that actually means that peroxidoxin 2 is showing an increased reduction of this particular cysteines at rest. And that really threw us because we hadn't expected that at all. Aging, as far as we were concerned, was actually associated with increased oxidation. So we went back into the literature and in fact to some of our own papers to see if we could find any more evidence that there was changes in reduction of these particular active cysteines at rest. So people will remember that when Brian with a proteomics approach to try and look at redox active cysteines. And this is the approach that he followed. What he did was actually used NEM, N-ethyl-maleimide, to block the thiols. And so he actually took, this is a schematic showing the NEM reacting with a thiol group here, and he just used the standard NEM to do that. He then reduced all of the oxidized cysteines, and he reduced it to then mean that we would actually generate further thiol groups, which could then be reacted with a heavy, so labeled NEM, so you could actually identify thiol groups that were either formed present as thiols or actually as disulfides or perhaps as mixed disulfides with glutathione and so on. And you could actually differentiate that between oxidized, reversibly oxidized and reduced thiol groups. And this was the paper that he published while he was with us in 2014. And so we've gone back to that data to try and understand the changes that occur with aging in lots of different thiols. So this is a figure from Neil Smith's paper from 2018. And what he's done here is actually to compare the cysteine redox state, so that is the amount of these different cysteines which are present in the reduced to oxidized form between old and adult. And what he highlights in the paper is the ones which have gone below the line. So they've changed in a oxidizing form, and this one here that I'm bringing is aconitase, which is a well-known protein present in mitochondria which is susceptible to oxidation and is being used as a marker, suggested as a marker of oxidation in aging. And you can see that of these, sorry, I've got the numbers in front of me here, but of these 75 peptides we've got here, there are about 13 of them which actually show increased oxidation. What the paper ignores is that 62 of them show increased reduction. And suddenly there's a realization that what we may have been missing is this idea of compartmentalization. Now, going back through the data, there's something like 45 proteins, with some of them which have got multiple peptides here. Of the 9 proteins which show oxidation, 5 of them are in mitochondria, 4 of them are elsewhere in the cell. But of the 20 proteins that show reduction, 19 of them present in the cytosol. So it looks like there's evidence that the cytosol is actually becoming more reduced, whereas the mitochondria becomes more oxidized with aging. So what's happening then with the redox status of our peroxydoxins? This is what we envisage, is that at rest in muscle, the peroxydoxins are sitting with a proportion of the potentially important active cysteines in a balance between reduction and oxidation, that when we activate the contractions, NADPH oxidase is activated, and we shift the balance into this redox signaling area. If we do oxidize it even more in some sort of toxic system, it might go into oxidative stress and damage. In old animals, we've got this increased stress towards reduction, particularly we think due to an increase in thyrodoxin 1, which is this protein which reduces peroxydoxins and maintains them in the reduced state. And that pushes this balance back to a reduced state. And then when the NADPH oxidase is activated, it doesn't actually reach the stage for activation of the signaling pathways. And that's actually what the data seem to show at the moment. So this is the hypothesis that we're working on at the moment, trying to understand. So what's the process? Well, this is the pathway you've seen before of NADPH oxidase activating peroxydoxins with activation. Eventually, we think of transcription factors. But alongside that, in another part of the cell, we've got the denervated motor axon terminal leading to an increased peroxide production by mitochondria. There then must be some direct redox signal to the nucleus, or perhaps a non-redox signal to the nucleus, because we don't see any effect in the cytosol. And that's been identified by others, and it's called mitonuclear communication. And it might involve things like, which I know years and so on. But there seems to be and it's modified transcription of proteins that regulate the oxidation site of these cytosol. So just trying to put it in terms of the global picture, in terms of what might well be happening and relating it back to innovation, this is what we think might well be going on. Throughout life, there's partial and small amounts of denervation where we lose axonal branches. That's just with exercise and everyday life. So schematically then, that's just shown as this branch being lost here, and this particular neuromuscular junction being oxidized. For a short period of time, that will induce a transient increase in mitochondrial peroxide production. We think that's there to stimulate axonal growth and sprouting, and you get reinnervation of this occurring, probably from sprouting from another axon or another axons of another motor unit. That peroxide will induce a minor amount of oxidative damage, and it will reduce short-term adaptations to reduce the risk of oxidative damage. But of course, that's only one of many, many events such as this which occur throughout our life. So it's not just this first one, we get further denervation and reinnervations, further denervations and reinnervations. And each one of these builds up. The proposal is to a chronic increase in oxidative damage markers, because there's little bits of oxidation occurring over time, which is what we see. But perhaps more importantly for our hypothesis here, they also lead to chronic adaptations to prevent further oxidative damage. And those adaptations involve some cytosolic adaptations. And that's actually what leads to this shift in the balance of the cytosolic proteins that we're seeing here. So that's our hypothesis. That's what we're trying to figure out at the moment. While we're validating. It obviously changes the way that we think about trying to restore these adaptations, this ability to respond to exercise. Antioxidants can't work. When you think about it, they might potentially make the system work. Targeting antioxidants might have some effect, but potentially they may influence this system. Maybe we should be looking to influence specific proteins which are driving these cysteines back into the reductive state. So this is the end of my talk. I've talked about how peroxidoxins act as effectors of hydrogen peroxide signaling. I've talked about the role of motor neurons and denervations in muscle mitochondrial redox homeostasis, this idea of compartmentalization and what that means to what's going on with aging. Just to say thank you to the group. I'm afraid it's an old slide. So not everybody's involved here, but I think I've mentioned most people as I've gone along, particularly the postdocs, Natalie, Caroline and Claire, who have been heavily involved. Kay had been working with Natalie very much on the NIH-driven work. Aphrodite and Vasiliki and Mattia very much involved in the redox adaptations to this. And I, of course, want to acknowledge Anne, who's been working with me for a long time, is absolutely key to all of the work that I've been describing here. And then these are our collaborators. Michigan and Oklahoma have been incredibly important for us. And Brian in Galway now has really played a very important part in this role. And I thank you all for your time. And I hope you can hear me most of the time.
Thank you very much, Malcolm. That's excellent and very interesting. You want to come back on, so we can perhaps see you again?
Sure.
There are a couple of questions. Roger, would you like to ask your question? Roger, you'll need to unmute yourself if you want to ask your question.
Sorry, I was in the lab. I've kind of got gloves on at the minute. I was wondering if you'd looked at, Malcolm, the effect of reinnervation on the redox status of the muscle, because if you reinnervate the muscle, in theory, the motor drive is recovered, but there's not a recovery of sensory drive. So that might help unpick the kind of driving signal to the muscle.
Yeah, we haven't done any.
Sorry, Malcolm, we've lost you totally. Can you try again? Can you start again?
Formal reinnervation experiments, but what we have done.
Oh, Malcolm, we keep losing you. Sorry, Malcolm, can you start from the beginning again?
So we haven't actually done any formal reinnervation experiments. So in other words, taken a denervated muscle and reinnervated it. But what we have done is the crush experiments that Caroline has done, where she's crushed the peroneal nerve and she's been able to follow the time course. And in that situation, then once the nerve repairs after the crush, the redox status of the muscle reverts as well.
Okay, thank you. Anne asks if TRX is key to recycling of the active PRX dimer. Is it the rate limiting, and what happens to the levels of TRX with age if you knock it out?
Okay. Thanks, Anne. I don't think I can answer either of those.
That's a friend for you, isn't it?
So the answer is yes, it's probably rate limiting. Don't think anyone has looked at this process with knockout of thyroidoxins. There's some redundancy between thyroidoxin 1 and thyroidoxin 2, but both have been knocked out and shown to reduce lifespan in animals. I think that's probably all I know.
and Rob asks, sorry, does it, sorry, Rob, would you like to ask your question? I don't understand it. Rob?
Hi, Malcolm. So basically my question is, I may have completely missed this, but you rescued one expression in your neurons, right?
Yeah.
So that seemed to rescue the redox state of the muscle fibers.
That's correct, Rob. Yeah.
How does that work? Because you think it's in the wrong cell. Does it
Yeah, it's
diffuse
in the
or
wrong
anything
cell.
like that?
Yeah. So it's because the actual SOD1 isn't doing very much in the muscle fibers at all, Rob. What happens is that the knockout of SOD1 in the nerves leads to these responses in the muscle mitochondria, this vast generation of peroxides. And so actually what happens is that when you rescue the SOD1 in the nerve, you actually stop the muscle fibers generating this big increase in peroxide. So it actually rescues the redox status of it. So I only briefly mentioned it, but a muscle-specific knockout has very little effect. You get no effect on mitochondrial peroxide generation with the muscle-specific knockout. It's all the muscle mitochondria responding to what the nerve's doing.
Okay, thanks.
So Malcolm, I've got a question which may be a rather naive one. You talked at the very beginning about the very different levels of peroxide in two different parts of the cell.
Yeah.
So one at nanomolar and one at micromolar, is that right?
Yeah, that's correct.
So,
well, extracellular is micromolar and intracellular.
Extracellular micromolar, but intracellular nanomolar.
Yeah, that's right.
So how is so if you've got one, so you're saying that the intracellular produces more extracellular peroxide, is that right? So how does a very low level produce much change into a very high level?
No, Rachel. The intracellular doesn't actually produce from muscle at least, doesn't produce the extracellular peroxides at all.
Oh, sorry.
I mean, what we're talking about is the process intracellularly where we're dealing with nanomolar concentrations. And but what everyone's done in the past is they've used micromolar because that's what they know happens extracellular, if you measure extracellular levels. So most of us in the lab will use micromolar concentrations of peroxides, and that's where the problem has occurred. That's actually meant we're dealing with alternative pathways. We're not dealing with these ones which
I see. I'm sorry, I misunderstood that. Any other questions, anybody? Well, thank you very much, Malcolm. Really interesting to see it all put together. And we could hear you most of the time. We lost a little bit, but very generally, the message came over very, very loud and clear. Thank you very much. And thanks, everybody. We'll see you again next week.